Okay, Aaron, thank you very much. It was always a pleasure to hear you. And I think, you know, it's amazing how cells are clever with this specificity. And we talked a lot about targeting, but the cells are doing it all the time and much more efficiently than we can dream of. I'll open the session for questions. Please.
Thank you so much for the excellent presentation. I was wondering if these ubiquitin ligases could be used as therapy against oncogenes?
Yeah. Now there are ways, I cannot go into it, chemists are developing it. Most of the diseases of the ubiquitin system are loss of function. Mutation, take for example Engelmann syndrome, which is a neurodevelopmental disorder of children. I didn't have time to go. Like many diseases. But there are now mechanisms to activate. Well, there are at least potential mechanisms to activate. It turns out that the ubiquitin ligases, the question was whether we can use ubiquitin ligases as a therapy. And the answer in principle is yes, but time has not come yet. But the principles are laid down, mostly in neurodegeneration in order to remove aggregated proteins. It turns out that the ligases are very promiscuous. As much as we think they are specific, all what we need to do is to bring them close to the substrate, to the ligase close to the substrate. And then it doesn't matter whether the ligase was created for something else. Any substrate that will come close, it will chew it down. So the mechanism, the question is how to do it. There are kinds of ways to do it. And people are starting mostly by attacking neurodegenerative disease in order to dissolve aggregated proteins like polyQ expanded proteins in Huntington or spinocerebellar ataxias, and even in Alzheimer, in tauopathies and so on and so forth. So they want to use the system as a general platform to trap proteins, but we are still not there. The chemical principle is there, but not the therapy.
Thank you.
So it's inspiring as always. You rightly mentioned the...
Where are you? Oh, yes.
You very rightly mentioned the similarity to phosphorylation. And one thing that always puzzles me is the fact that many processes of tagging in biology are very simple, like phosphorylation. However, in the case of ubiquitin, it's a 76 amino acid tag, and with the polyubiquitin, it's hundreds of amino acids. It seems to be not efficient energetically, but yet, as we see with the SOMO and other families, probably it is. So it is coincidence, or you need to have such a costly label?
It's even more costly than you think it is. Much more costly, because phosphorylation costs you one molecule of ATP. Ubiquitination costs you at the beginning two, because the ATP is cleaved between the alpha and the beta. So you lose two ATPs, and then on a chain, it's double the number. And then what's really amazing, that ubiquitin itself is degraded partially with the substrate. So you are losing a whole protein in order to degrade. So you are taking a small molecule, let's say a molecule of 15,000, you piggyback it with 500,000 chain of ubiquitin, and half of this 500,000 is degraded. Actually, the absurd, well, I don't dare to call God an absurd, or evolution an absurd, depends on whom you believe. The real calculation shows that in degradation of a protein by the ubiquitin system, you invest at least, if not more, the amount of energy that you invest in the synthesis of the same very protein, which is amazing, just shows you how much you need to invest in control. Now, the comparison between phosphorylation and ubiquitination, I cannot make it. They talk to one another, and they are both. Phosphorylation is much simpler, you know, it's phosphate and 3 amino acids. Ubiquitination is an endless language. The potential is huge, and it serves numerous functions. I cannot tell you that more functions than phosphorylation. So it's hard to, to imagine why such a cumbersome language has evolved, but there is actually, I don't know, any cellular process in which ubiquitination is not involved, but the same for phosphorylation. So, it's another layer. We cannot really compare, but the cost is huge, and the language is far more complicated.
Why does the system not more effectively manage these profibrillar structures that underlie, for example, Parkinson's and Alzheimer's?
Yeah. It's an excellent question why the system doesn't manage aggregated proteins. It seems that aggregated proteins are chewed but very slowly and from the periphery, and they kind of block the proteasome. They kind of clog it. So you find the active ubiquitin system and you find an active proteasome that is being clogged by aggregated proteins. And the idea is to really augment the process and to peel it off. The protein, you know, it's like a hard egg. It's very difficult to get in, and you have to peel it off from the periphery. And it seems that if you stop aggregation, so there are ways to do it. If you stop aggregation, the aggregates will go away. The problem is that it's a continuous process. So there is a competition between aggregation and dissolution of the aggregate. So if we shall manage somehow to stop aggregation, people try to do it by overexpression of chaperones and so on, then the ubiquitin system will manage. But it's a matter of pace. But you're right.
So the peptides which are coming out from the proteasome, how big are they and what happens with them?
Well, the peptides that are coming are between 10 to 20 amino acids, and it's a very good question because these peptides are being captured by the MHC class 1 molecules, and they are presented to the CD4 cells. Now, so the ubiquitin system, among its many functions, this is like a side function because it's a side it's a final product, is also the main arm of the recognition by MHC class 2 antibodies, MHC class 1 molecules. So all the foreign proteins, let's say the ubiquitin system doesn't distinguish between proteins. So let's say that you have a viral infection like flu or any viral infection, the ubiquitin system will generate peptides of flu. These peptides of flu will be presented to MHC class 2 molecule, to MHC class 1 molecules, and will elicit dissolution of the presenting cell. So by making the antibodies, rather than by making amino acids, the ubiquitin system serves as the main arm of the immune system. I didn't mention it, but yeah, it's a very good question. And then the peptides, you know, for presentation on MHC class 1 molecule, you need little fraction. The rest are being degraded by amino and carboxypeptidases.
So on the proteasome, I just want to sort of push that a little bit further. Have you started to work with the modulation of the rate of the proteasome activity as opposed to completely blocking it? Or so you could actually use that, going back to the MHC2 peptides that are going to be presented, you could actually use this to induce a higher immunogenicity to bring the viral response up much faster for vaccines and things. Okay. As well as in neuro, you could use it to decrease the amount of protein that's being produced that may hold the brain in the three-dimensional space.
It's an excellent question. The proteasome doesn't seem to be limiting. You know, the proteasome is 2% of the total cellular proteins. It's a huge fraction, 2% of the total cellular proteins. There is a fraction of the proteasome that is called the immunoproteasome. So if you put interferon into cells, the proteasome exchanges some of its proteolytic beta units into units that will cleave in sites that are more ideal to generate the right antigen. But even without these beta units, it's not so awful because then there is an ER-associated protease, which is called ERAP. So even if you are making a longer peptide, once the peptide crossing the ER membrane on the way to the MHC class 1 molecule and the beta-2 microglobulin, ERAP will cleave it to the right size, but it takes more time. But the proteasome can adapt itself by immunomodulators like interferon. I don't think that we need more proteasome. The problem with neuro, as I said, is looks like inhibition of the proteasome by aggregated proteins. And if we can somehow release this blockade, then I think the proteasome will act because it's an active proteasome. But you're right about the immunoproteasome and its induction during infection in specific cells.
So, I would like to follow on this question about the MHC. Don't you think that rather than induce an immune response, this will be a wonderful mechanism to induce immune tolerance to our self-proteins? because you don't have any danger signal. You have a continuous renewal of all these proteins. And this could even be used for therapeutical purposes.
So what you are arguing is what? That it would be better that the proteasome will degrade the proteins altogether?
No. So what I'm saying is that these peptides that they are produced by the ubiquitin system, they rather than induce an immune response, they should induce immune tolerance. They are self-proteins, and they are presented in a context that there is no danger, there is no infection. These are our self-proteins, and probably this is a very important mechanism to keep us under control, because all our T cells are autoreactive. There is no...
But you are looking, you are right in principle, yeah. I'm not an immunologist, so I didn't think about it. It's absolutely a provoking idea to think about it. But don't forget that the ubiquitin system, for example, activates NF-kappa B. So you have activation of the immune response by stimulating immune modulators like interleukin on the other side. So then you have to control also NF-kappa B. So I think that it's more complex than what you suggested. If we would be only limited to the MHC class 1 molecules, then you are right. But I think that it's more complex. But I have to think about it. I must admit, you caught me in a surprise. So, but it's a very good one. It's an absolutely good one. But then you have the CD4, so you can contain the immune system is working there because you need little of presentation in order to lyse the presenting cells. So it's quite efficient.
Okay, I think
Thank you.